I'm an RNA biologist, and what that means is I study an intermediate molecule in what's known as like the central dogma of molecular biology. So in the lab, major focus is cancer and cancer biology. So when I started my WRF Fellowship, at the time we had really great technologies to measure and look at all of the RNAs in the cells. So we could come up with this great catalog of all the RNA in a normal cell and all of it in a cancer cell. But the big gap that existed at the time was we had no way of actually functionally studying each of these. So we could see them, but we couldn't actually delete any of them and ask what happens. The WRF supported my work to develop this technology so that we could then take cancerous cells and we could see that they're producing all of these strange RNA molecules. Now we're going to use this technology, we're going to remove one, and we're going to ask what happens. And the different things that can happen are things like, if we remove this, does the cancer cell start to slow down in its growth? Oftentimes these RNA molecules are associated with other things, like therapeutic resistance. So if we have some drug which works really well for certain patients, but we add it to this cell and there's no effect, we want to ask questions like, wait, but what if we remove this RNA? Does that cell now become sensitive to this drug? So in that instance, this is a way for us to increase the proportion of patients who are candidates for therapy. Through the support of the WRF, I was able to design this technology in a way where it's not just pigeonholed to be specific to the one sort of disease or context I study, but now it's able to help any scientist who's studying how RNA is associated with their biological system or disease of interest. I benefited a lot from just the sort of environment that the WRF created to think about science. So even from the beginning, it was, here's this proposal, let's make this a reality.